Hi everyone, my name is Paola Dama. I'm founder and CEO of NAMina Bio. NAMina Bio is building a translation architecture network focused on integrating the new approach methodologies into more predictive, human-relevant workflows for therapeutics development, disease modeling, biological barriers, and translational decision making. For us, the key word is integration. We know that today the field has access to powerful technologies, organoids, 3D bioprinting, microphysiological systems, dynamic culture platforms, multiomics, imaging, and AI-assisted analytics. But this technology often remains fragmented. The real translation value emerges when they are connected around a clear biological question. So my focus today is to show how NAMina can integrate dynamic microenvironment and fluidic culture systems into broader translational workflows. So the central idea is simple. Dynamic culture becomes most valuable when it is connected to the right model, to the right readouts, and the decision-ready interpretation. At NAMina Bio, we organize advanced in vitro technology into pilot studies and translational programs. So the goal is not only to run an assay or generate a dataset. The goal is to create a structured workflow that can answer a specific translational question. For example, can a therapy penetrate a complex tumor microenvironment? Or does the response change under flow, perfusion, or oxygen gradients? Does a biologic barrier alter drug transport efficacy or toxicity? Can we detect the resistance earlier before it becomes visible in simple models? So there are not only technical questions, they are decision-making questions. This is why we combine three elements: human relevant systems, predictive data, and translational interpretation. So the human relevant system may be an organoid and 3D tissue barrier model or co-culture, a dynamic culture platform, or organ-on-a-chip system. The data may come from functional response, imaging, phenotype profiling, molecular analysis, or multiomics. But the key value is the interpretation, turning the data into something useful, such as a clear mechanism, therapy prioritization, biomarker hypothesis, regulatory aligned evidence, or next step recommendation. So NAMina is not positioned as a single technology company. We are building a framework to connect the right model, the right dynamic condition, and the right analytical layer for each context of use. Many promising therapeutic programs still fail because early models do not fully capture human biology. So this is especially important in complex areas such as oncology, inflammatory disease, barrier biology, or tissue-specific toxicity. Traditional models have been essential, but they have a limitation. Animal models do not always predict human outcomes, and static 2D systems are often too simplified, as we learned. So an even advanced 3D system can miss important physiological variables if they are maintained under static conditions. So for NAMina, the issue is not only model complexity. The deeper issue is whether the model generates evidence that can support a better decision. A system may capture structure but not adaptation. It may capture marker but not functional response. It may capture single time point but not how biology evolves under stress. This is why we need both better model and better evidence frameworks. We also need those evidence frameworks to be structured enough to support a better translation and regulatory decisions. So for NAMina, this is the opportunity to bring together human relevant biology, dynamic experimental systems, interpretable analytics, and context of use driven evidence generation early in development. So decisions are based on better context, better evidence, and stronger translational confidence. The slide summarizes the NAMina approach. We think about translational modeling as an integrated path with the three main layers. The first layer is the human relevant model. As we just said, the organoid 3D tissue. The second layer is dynamic experimental system. This is where technologies such IVTech, fluidic culture platform can provide an important bridge by adding controlled physiological context to advanced in vitro model. The third layer is decision-ready analytics. A model becomes truly useful only when the data can be interpreted in a way that supports the next step, continuing to prioritize, combine, optimize, or investigate mechanisms. At NAMina, we connect these layers into fit-for-purpose workflows rather than starting with the technology and asking, what can we do with it? We start with the biology and translational question. So what do we need to understand? What decision does the data need to support? Which model and dynamic condition are appropriate? Which readouts will make the result interpretable? So this is the difference between an isolated experiment and a translational program. So the key message is not single technology, but an integrated path to better evidence. Dynamic culture systems are important because they add the physiological context to advanced in vitro models, but from NAMina perspective, the value is not simply that we can add flow or perfusion. The value is that we can place those conditions inside a structured translational workflow. For example, in tumor microenvironment modeling, dynamic culture can help us to evaluate how architecture, extracellular matrix, stromal components, immune interactions, and exposure dynamics influence response. In barrier and interface studies, dynamic culture helps move beyond the question of whether a compound crosses a barrier toward understanding how transport, penetration, and response behave under more realistic conditions. And when dynamic culture is combined with imaging, functional readouts, and multiomics, we can begin to understand not only whether something works, but why it works, why it fails, and under which biological condition the response changes. So my message here is not that dynamic culture is only a better way to screen drugs. It is a way to generate richer biological context, and NAMina's role is to integrate that context into decision-ready translational evidence. This slide brings together the way NAMina structures human-relevant technologies into a translational workflow. So the main point is that these technologies are not used in isolation. Their real value comes when they are organized around a clear biological question and translated into decision-ready evidence. At NAMina, we start with the clinical biological question. Let's say, what disease context are we modeling? For example, model or biological system are we using? And most importantly, what decision does the data need to support? From there, we move into study design and model strategy. This is where we define the model architecture, the extracellular matrix, the level of biological complexity, and the assay plan. The next step is the 3D model generation, which may involve the organoids, bioprinted tissue, tumor-on-chip system, barrier models, or other advanced in vitro system. Then we add the dynamic physiological layer. This is where dynamic culture system can introduce flow, perfusion, oxygen control, exposure dynamics, and potentially multi-system context. This step helps move to the model from a static representation toward a more physiological, relevant experimental condition. Once the model is established under the right dynamic context, we proceed with the functional testing, such as therapeutic response imaging, immune interaction, barrier function, or phenotypic readouts. The next layer is a molecular computational analysis, where we can integrate genomics, transcriptomics, multiomics, and predictive analytics to better understand not only whether a response occurs, but why it occurs. Finally, the output is not just raw data. The output is a decision-ready translational report, mechanistic insight, therapy prioritization, biomarker hypothesis, human relevant evidence, and next step recommendations. So the workflow is designed to move from complexity to clarity. Dynamic culture gives us more physiological context. So NAMina's role is to integrate that context with the right model, the right readouts, and the right analytics, so it becomes useful translational evidence. So to make this platform workflow more concrete, I'll show one illustrative example where we apply this logic to mature tumoroids resistance profiling and extend it with a dynamic physiological layer. This slide shows an illustrative case study of how NAMina Bio can extend mature tumoroids profiling into a more dynamic, physiologically relevant workflow. This concept builds from our mature tumoroids work developed with the real research using live gel-based 3D tumor system to better capture tumor architecture, matrix interaction, and time-dependent drug response. So the key idea is that resistance is not always visible in simple models. In standard 2D culture, a compound may appear highly effective, but that can be an oversimplified snapshot because the cells are exposed directly to the drug without the architecture or microenvironmental constraint of a tumor system. When we move into an early 3D tumoroid, we begin to capture the architecture. The response may already shift because cells are no longer growing as a flat monolayer. Then in mature tumor stage, the model has more time to develop structural and biological features that are closer to the tumor behavior. This is where we may begin to see time-dependent adaptation and reduced sensitivity. The next step is the dynamic mature tumoroids. By adding flow, perfusion, oxygen, and nutrient gradients, a drug exposure dynamics, we can ask a deeper question, not only whether the tumor responds, but how that response change under more physiological conditions. The purpose is to increase biological realism step by step, from 2D, early 3D, to mature 3D, and then to dynamic mature tumoroids. The output is not just another dose-response curve. The output is a decision-ready interpretation, hidden resistance phenotypes, more realistic effective dose ranges, combination strategy insights, and biomarker hypotheses. So for NAMina, this is exactly the value of integration. Real research provides a strong biological foundation through mature tumoroid system, and NAMina builds on that foundation by integrating dynamic culture, functional readouts, and translational analytics into a workflow that can support better preclinical decision. To wrap up, our goal at NAMina Bio is to turn complex human relevant data into clear, confidence-building translational decisions. We are currently developing pilot programs that integrate advanced 3D models, dynamic culture system, functional readouts, and analytics into structured evidence packages. So if you are interested in exploring how this approach could support your own program, so please feel free to reach out to me directly, and thank you for your attention, and I look forward to the discussion and meeting you. Bye.